Welcome to the Deep Dive, where we plunge into complex topics to get you informed quickly and clearly. Today, we're tackling something really fundamental, but also, well, surprisingly dynamic. Our own genetic code. It's kind of a paradox, isn't it? DNA is supposed to be this super stable blueprint of life, yet it's constantly in flux. Okay, let's unpack this. Our mission today is to really dive into the hidden world of gene mutations, DNA repair, and this amazing thing called transposition. We'll explore how these genetic changes happen, how our bodies, heroically fight back, and their huge impact on everything from evolution to disease. It's a journey into how stability and change are sort of dancing together in our very cells.
Exactly. And it's crucial to remember that these shifts, these mutations, while sometimes, damaging, they're also the raw material for adaptation, for evolution. They generate the variation that life needs. But on the flip side, they're also the source of many genetic diseases and, of course, cancers. We'll be drawing heavily on the insights from Essentials of Genetics, the 10th edition, to guide us through this complex landscape.
Great. So let's start right at the beginning. What is a mutation, really, at the gene level?
Simply put, it's an alteration in the nucleotide sequence of an organism's genome. Think of it like a typo in that incredibly long genetic script. It could be a single letter, a base pair changing, or maybe a letter being deleted, or an extra one inserted.
Okay, a change in the sequence. But how much impact can one little change have?
That's the key thing. The effects can range from, well, absolutely nothing to something really catastrophic. It depends less on the size of the change and much more on where it happens. Is it in a crucial part of a gene that codes for protein? Or is it maybe in a non-coding region, like an intron, or something that controls the gene, like a promoter or an enhancer? Or maybe it affects how the message gets spliced together. Location is everything.
Right, location, location, location. So how do scientists break these down? Like, what are the different types of these molecular changes?
Okay, we can classify them first by the molecular change itself. The most common ones you hear about are point mutations, or base substitutions. That's where just one base pair swaps for another. And even within those single letter swaps, there are different outcomes for the protein. You can have a missense mutation. One letter changes, and that results in a different amino acid being put into the protein chain.
Okay, so the meaning changes slightly.
Right. Then there's a nonsense mutation, which is often more dramatic. A single letter change creates a stop signal, a stop codon, right in the middle of the gene.
So it just cuts the protein short.
Exactly. Protein production halts prematurely. And then there's the silent mutation. The letter changes, but remarkably, the amino acid stays the same.
How does that happen?
It's because of the redundancy in the genetic code. Multiple three-letter codons can actually code for the same amino acid. So the DNA changes, but the protein sequence doesn't. It's stealthy.
Huh. And I remember reading about transitions and transversions within these point mutations.
Yeah, that's just a finer classification. Transitions are when a purine base swaps for the other purine, like A for G or G for A, or a pyrimidine swaps for the other pyrimidine, C for T, T for C, sort of like swapping within the same chemical family. Transversions are when a purine swaps with a pyrimidine or vice versa, like A for C or G for T, a cross-family swap.
Got it. Okay, so that's changing single letters. What about adding or removing letters, insertions or deletions?
Right, those are the indels insertions or deletions. And these can be really disruptive, especially if the number of bases added or lost isn't a multiple of 3. Think of that classic analogy, the cat saw the dog. If you delete the C from cat, the whole reading frame shifts.
Right, you get the ATs, RDTHEGOG, total gibberish.
Exactly, that's a frameshift mutation. Adding or losing even a single letter throws off the entire 3-letter reading frame downstream from the mutation. This usually leads to completely garbled proteins that are often truncated and non-functional.
And I guess the earlier in the gene it happens, the worse it is.
Generally, yes. Much more of the protein sequence gets altered.
Okay, so that's classifying by the change. What about classifying by the effect on the gene's function?
Good question. We often talk about loss-of-function mutations. These, as the name suggests, reduce or completely eliminate the function of the gene product. If it completely eliminates function, we might call it a null mutation. Most of these tend to be recessive, meaning you usually need 2 bad copies to see the effect, because 1 good copy can often compensate.
But you mentioned sometimes loss of function can be dominant. How does that work? Seems odd.
It does sound counterintuitive, but it happens. One way is through dominant negative mutations. Imagine a protein that needs to pair up with an identical copy of itself to work a homotomer. Now, if one copy of the gene has a dominant negative rotation, it produces a faulty protein subunit. This bad apple can actually interfere with the normal protein produced by the good copy, poisoning the whole complex.
So one bad one ruins the bunch.
Pretty much. Another way is haploinsufficiency. This is where having just one fully functional copy of the gene simply isn't enough to produce the normal amount of gene product needed for a healthy state. Marfan syndrome is a classic example. It's caused by a loss-of-function mutation in just one copy of the FBN1 gene. One good copy just doesn't make enough fibrillin-1 protein for normal connective tissue.
Wow, okay. And the opposite, gain-of-function.
Right. Gain-of-function mutations are where the gene product gets a new function, or maybe its existing function is enhanced or inappropriately activated. These are often dominant because the new or overactive function makes itself known even with the normal copy present.
And suppressor mutations, the undo button.
Yeah, suppressor mutations are fascinating. It's a second mutation at a different site that counteracts or alleviates the effects of the first mutation. It doesn't necessarily fix the original mistake, but it compensates for it somehow.
Okay, and we can also see mutations manifest in all sorts of ways, phenotypically, right? Like visible changes.
Absolutely. They can be visible, think altered eye color in flies or coat color in mice. They can be nutritional, where an organism loses the ability to make a vital nutrient. Or biochemical, like the altered hemoglobin in sickle cell anemia. Some affect gene regulation, changing when or where a gene is turned on or off. Some are tragically lethal, like in Tay-Sachs or Huntington disease. And others are conditional, only showing their effect under certain conditions, like the temperature-sensitive allele giving Siamese cats their distinctive pointed pattern. The enzyme only works in cooler extremities.
That Siamese cat example is always so clear. And the final classification is location within the body, somatic versus germ-line.
Correct. Somatic mutations occur in your regular body cells. They happen after conception, aren't passed on to your kids, and their impact depends on when and where they occur. If dominant, or if they happen early in development affecting many cells, they can have significant effects, including contributing to cancer.
But germ-line mutations, those are the inheritable ones.
Yes. These occur in the cells that produce sperm or eggs. These mutations can be passed down to the next generation. That's why a recessive germ-line mutation might hide unseen for generations, only appearing when, by chance, two carriers have offspring who inherit two copies of the mutated allele.
Right, that gives us a really solid map of what mutations are. Now, the big question, where do they actually come from?
You mentioned spontaneous versus induced.
Exactly. Let's start with spontaneous mutations. These are changes that just happen. They arise naturally from normal biological or chemical processes inside the cell, often during DNA replication itself, but also from other internal factors. No specific external agent is involved.
So like unavoidable errors, how often do they happen?
The rates are generally incredibly low, which speaks volumes about the fidelity of DNA replication and repair. But intriguingly, these rates vary quite a bit between different organisms, bacteria versus corn versus mice, for example. Why such variation exists is still an area of active research.
And what about us? Humans. Ah, yes. There was a really significant study done in Iceland back in 2012. They sequenced the genomes of parents and their children, and they found that, on average, a newborn has about 60 new mutations that weren't present in either parent's genome. 60.
60 new changes in every baby, wow.
Yeah, and here's a really interesting finding. The father's age plays a big role. A 20-year-old dad contributes around 25 of those new mutations on average, but a 40-year-old dad, he contributes about 65.
Whoa, that's a big difference. Why?
It's thought to be because the male germ cells, the sperm precursors, go through continuous cell divisions throughout a man's life. More divisions mean more opportunities for DNA replication errors to creep in.
And the mother.
The mother's contribution is much lower, around 15 new mutations, and it doesn't seem to increase significantly with her age because egg cells are formed much earlier and don't divide continuously in the same way.
That's fascinating. So are these 60 new mutations mostly harmless?
Mostly, yes. The study found most were in non-coding regions of the genome. Only about 73, on average, were within exons, the protein-coding parts. And estimates suggest maybe only 10% of those actually lead to negative phenotypic effects, so perhaps around 6 potentially deleterious effects per generation. Still, it adds up over evolutionary time.
And you mentioned somatic mutations are even more frequent.
Oh yes. Estimates are that somatic mutation rates can be 4 to 25 times higher than germline rates. This constant accumulation of mutations in our body cells throughout life is a major factor in the development of cancer.
Okay, so replication errors are one source of spontaneous mutations. What else is happening inside our cells?
Well, DNA replication isn't perfect. Sometimes the polymerase just grabs the wrong nucleotide. Proofreading catches most, but not all. Then there's replication slippage. Especially in areas with repeated sequences, the DNA strands can kind of loop out or misalign, causing the polymerase to stutter, adding or deleting copies of the repeat. This is actually the mechanism behind diseases like Fragile X and Huntington's.
The repeats expand.
Exactly. Another issue is tautomeric shifts. The DNA bases themselves can temporarily flicker into alternative chemical forms, or tautomers. If this happens right when the DNA is being copied, it can cause the wrong base to be pair opposite it, leading to a permanent point mutation in the next round of replication.
Chemical instability.
And sometimes bases just fall off. Depurination is the loss of a purine base, an A or a G. This happens thousands of times per day in a typical mammalian cell. It leaves a gap, an apurinic site, and when the polymerase encounters it, it might just guess and slot in a random base, often an adenine.
Thousands a day. That's incredible.
It is. Then there's deamination, where a base chemically changes. A common one is cytosine losing an amino group and turning into uracil, which normally belongs in RNA, not DNA. If not repaired, it leads to a CG pair becoming a TA pair. Adenine can also be deaminated.
And finally, oxidative damage.
Yes, oxidative damage. Just the process of breathing and using oxygen creates reactive oxygen species free radicals as byproducts. These are highly reactive chemicals that can attack DNA, causing over 100 different types of base modifications. It's constant wear and tear.
Phew. It really is amazing our DNA holds up as well as it does.
Okay.
Okay, so that's spontaneous. What about the induced mutations from external factors?
Right, induced mutations. These are caused by environmental agents or mutagens. We can group them into chemical mutagens and radiation.
Let's start with the chemicals.
Okay. Some are base analogs. These are molecules that look structurally similar to normal DNA bases. 5-bromouracil, or 5-BU, is a classic example. It looks like thymine, so it can get incorporated into DNA opposite adenine. But its structure is a bit unstable, and it can shift into a form that pairs with guanine instead. This causes AT pairs to eventually become GC pairs.
Sneaky mimics.
Then you have alkylating agents. These chemicals donate alkyl groups like ethyl or methyl groups to the DNA bases. This alters their structure and changes their base pairing properties, leading to transition mutations. Mustard gases used in chemical warfare are potent alkylating agents, as is EMS, a common lab mutagen.
Nasty stuff. What else?
Intercalating agents. These are flat molecules that can slip themselves or intercalate right in between the stacked base pairs in the DNA double helix. This distorts the helix structure. When DNA is replicated, this distortion can cause the polymerase to slip, leading to insertions or deletions, frameshift mutations. Acridine dyes are examples.
They wedge themselves in.
Yep. And finally, adduct-forming agents. These substances react covalently with DNA, attaching themselves to it and forming bulky additions called adducts. These adducts interfere with replication and repair and can lead to mutations. Acetaldehyde from cigarette smoke and heterocyclic amines, HCAs, formed when cooking meat at high temperatures are examples linked to cancer.
Okay, chemicals covered. What about radiation?
Radiation is a major mutagen. Ultraviolet, UV light, particularly the UVC and UVB wavelengths from sunlight, is strongly absorbed by DNA bases. This energy can cause adjacent pyrimidine bases, especially thymines, on the same strand to covalently link together, forming pyrimidine dimers, most commonly thymine dimers. These dimers create a bulge that distorts the helix, blocks DNA replication, and can lead to errors if the cell tries to replicate past them.
So sunscreens are definitely a good idea.
Absolutely. Then there's ionizing radiation. This includes things like X-rays, gamma rays, and cosmic rays. It's much higher energy than UV. It penetrates deeply into tissues and can cause damage in a couple of ways. It can directly hit the DNA, but more often it ionizes water molecules in the cell, creating highly reactive free radicals. These radicals then attack the DNA, causing various types of damage, including chemical modifications to bases, and most dangerously, breaks in the DNA backbone, including double-strand breaks.
Double-strand breaks sound really bad.
They are the most severe type of DNA damage, as they can lead to chromosome loss or rearrangements like deletions, duplications, inversions, and translocations if not repaired correctly.
Where does most of our ionizing radiation exposure come from? Is it mostly man-made sources?
Actually, no. It's estimated that less than 20% of the average person's exposure comes from human activities like medical X-rays. The majority comes from natural sources: radon gas seeping from the ground, cosmic rays from space, and natural radioactivity in soil and rocks.
Huh, that's surprising. Okay, so our DNA is constantly under siege, both from within and without. This leads us directly to the impact on health. Single gene changes causing serious disorders.
Exactly. While many common diseases are complex, involving multiple genes and environmental factors, there are thousands of inherited disorders caused by mutations in single genes. The online Mendelian Inheritance in Man, or OMIM database, catalogs these, and the types of mutations involved are revealing. About 30% of known disease-causing mutations are nonsense mutations, creating those truncated proteins. Missense mutations are also very common, altering protein function, frameshifts too. And interestingly, about 15% of point mutations that cause genetic diseases don't affect the protein sequence directly, but instead mess up mRNA splicing by hitting those crucial sequences at the boundaries between exons and introns.
Affecting how the message is put together.
Precisely. A great example that showcases the variety of mutations causing a single disease is beta-thalassemia.
Right, the blood disorder.
Yes, it's an inherited autosomal recessive condition, very common worldwide. It results from reduced or absent hemoglobin beta chains, leading to anemia and other symptoms. What's fascinating is that it's caused by mutations in one gene, the HBB gene. But there are around 400 different mutations known in that single gene that can cause thalassemia. Most cases actually stem from about 20 more common mutations, but they affect virtually every part of the gene and its expression. The promoter region upstream, the start codon, exons, introns affecting splicing, the polyadenylation signal, even large dilutions removing the whole gene. It really highlights how vulnerable genes are.
A single gene. Hundreds of ways to break it. That's sobering. But thankfully, our cells aren't defenseless, right? They fight back with DNA repair systems.
Absolutely essential systems. Life as we know it wouldn't exist without them. The mutation rates we actually observe are the net result of the damage rate minus the repair rate. These systems are constantly scanning and fixing.
What's the first line of defense?
That's the proofreading function of DNA polymerase itself during replication. It has a sort of backspace key, a 3 to 5 foot exonuclease activity. If it adds the wrong base, it can often detect the mismatch, pause, snip out the wrong base, and try again. This improves replication fidelity by about 100-fold.
Pretty good spell checker. But some errors still get through.
Yes. And for those, the next line of defense is mismatch repair, MMR. This system acts like a post-replication cleanup crew. It scans newly synthesized DNA, recognizes mismatches or small insertions, deletions that proofreading missed, figures out which strand is the new and therefore incorrect one, cuts out the faulty segment, and replaces it correctly using the original template strand as a guide.
And if MMR itself is faulty?
That has serious consequences. Mutations in human MMR genes like HMSH2 and HMLH1 are strongly linked to certain types of cancer, particularly hereditary non-polyposis colon cancer, HNPCC, also known as Lynch syndrome. These defects lead to what's called genome hypermutability, a massive increase in the mutation rate across the entire genome, greatly increasing cancer risk.
Okay, so proofreading and MMR handle replication errors. What about damage that happens after replication or damage that stalls replication?
For situations where the replication machinery encounters damage it can't immediately fix, like a UV-induced dimer, there are other systems. One is postreplication repair. Here, the polymerase might skip over the lesion, leaving a gap. Then a system involving the RET-A protein in E. coli can mediate a sort of recombinational exchange, borrowing the correct sequence from the undamaged sister strand to fill the gap. It's a form of homologous recombination repair.
Using the backup copy. Smart.
And in bacteria like E. coli, if the DNA damage is really extensive and overwhelming, they can activate the SOS repair system. This is truly a last resort. It involves inducing the expression of about 20 different genes whose products allow replication to proceed even across damaged templates. The catch is that this process is highly error-prone. It often inserts random bases opposite the lesions.
So it saves the cell from dying, but at the cost of introducing new mutations.
Exactly. It's mutagenic, but better than certain death for the cell.
Now, what about that cool light-activated repair? Photoreactivation.
Photoreactivation repair, yes, it's elegant. Many organisms, from bacteria to plants to some animals, have an enzyme called photolase. When activated by visible blue light, it directly binds to UV-induced pyrimidine dimers and breaks the covalent bonds, perfectly restoring the original bases.
Direct reversal. Why don't we have that? We get plenty of sun exposure.
That's a great evolutionary question. Placental mammals, including humans, seem to have lost the gene for photolase during evolution. We rely entirely on other mechanisms, primarily excision repair to deal with UV damage. Why we lost it is unclear. Perhaps the other systems became efficient enough, or there was some trade-off.
So we rely on cut and paste instead, excision repair.
Exactly. Excision repair is a major pathway in almost all organisms. It's a more general strategy. The basic steps are: 1. Recognize the damage or distortion. 2. Enzymes clip out the damaged section, including some surrounding bases. 3. DNA polymerase fills the gap using the intact opposite strand as a template. And 4. DNA ligase seals the final nick in the backbone.
And there are different types of excision repair.
Yes. Two main types. Base excision repair, BER, primarily deals with single damaged or inappropriate bases like uracil in DNA or chemically modified bases caused by alkylation or oxidation. Special enzymes called DNA glycosylases recognize and remove just the damaged base, creating a site without a base. Then other enzymes cut the backbone, remove the sugar phosphate, and polymerase and ligase fill and seal.
Targeted removal of just the bad base.
Right. Then there's nucleotide excision repair, NER. This pathway handles more bulky lesions that distort the DNA helix significantly, like those UV-induced pyrimidine dimers we talked about, or large chemical adducts. NER enzymes recognize the distortion, make cuts on either side of the lesion, remove a larger chunk of the single strand, around 30 nucleotides in humans, and then polymerase and ligase do their jobs to fill the gap.
And NER is the system that's defective in xeroderma pigmentosum, XP.
Precisely. Xeroderma pigmentosum, XP, is a rare, devastating autosomal recessive disorder. Individuals with XP have extreme sensitivity to sunlight because their NER pathway is defective. They can't repair UV-induced DNA damage properly. This leads to a dramatically increased risk, like 2,000-fold higher, of skin cancers, often starting in childhood, as well as premature skin aging and often severe neurological problems.
And studying XP revealed something important about the complexity of repair, right?
Absolutely. When researchers fused cells from different XP patients together, forming heterokaryons, they sometimes found that the fused cell could carry out normal NER. This meant the patients had defects in different genes required for the pathway. This complementation analysis identified at least 7 different NER genes, XPA through XPG, involved in human NER. It showed just how complex this repair pathway is, requiring multiple proteins working together.
Okay, BER and NER handle base damage and bulky lesions. What about those really dangerous double-strand breaks?
Double-strand break, DSB repair, is critical because DSBs can lead to chromosome fragmentation and loss. Eukaryotes have 2 main pathways for this. The first is homologous recombination repair, HRR. This is a high-fidelity pathway because it uses the undamaged sister chromatid, which is usually available after DNA replication in the late S or G2 phases of the cell cycle, as a template to accurately repair the break. It essentially copies the missing information from the identical sister molecule.
Accurate, but only works when there's a sister chromatid available.
Correct. The other major pathway is non-homologous end joining, NHEJ. This pathway is active throughout the cell cycle, especially before replication in G1 when no sister chromatid is available. NHEJ basically just takes the 2 broken ends and sticks them back together. It's faster, but it's inherently error-prone. Often, some nucleotides are lost or added at the break site during the processing before joining. And sometimes, it can even mistakenly join the wrong broken ends together, leading to chromosomal translocations. Proteins like BRCA1, famous for its link to breast cancer susceptibility, play roles in DSB repair choices and execution.
So, a quick fix, but potentially messy. It's just incredible, this constant molecular surveillance and repair going on. Now, shifting gears slightly, how do scientists actually test if a chemical might cause these mutations in the first place? How do you screen for mutagens?
That's where the Ames test, developed by Bruce Ames, is really fundamental. It's a clever and relatively simple bacterial assay used widely for decades to screen chemicals for mutagenicity.
How does it work?
It uses special mutant strains of the bacterium Salmonella typhimurium that have a mutation making them unable to synthesize the amino acid histidine, His. They can only grow if histidine is provided in their culture medium. The test essentially asks, can the chemical we're testing cause these His bacteria to mutate back to the wild-type state, his plus, allowing them to grow without added histidine. This back mutation is called reversion.
So you expose the bacteria to the chemical and see if they regain the ability to make histidine.
Exactly. You plate the bacteria on a medium lacking histidine, add the test chemical, and count how many colonies grow. A significant increase in colonies compared to a control plate without the chemical indicates the chemical is mutagenic. And crucially, the test often includes an extract from rat liver enzymes. This is important because many chemicals aren't mutagenic themselves, but they become mutagenic after being metabolized by enzymes in the liver, just like in our bodies.
Mimicking human metabolism. Does it work well?
Remarkably well as a preliminary screen. There's a strong correlation, something like over 80%, between compounds that are known carcinogens, cancer-causing in animals, and those that test positive in the Ames test. It's widely used in industry and drug development as a rapid initial assessment of mutagenic potential.
Okay, a powerful screening tool. Now, for our final topic, let's talk about something really wild. The jumping genes, transposable elements, TS.
Ah, yes, TEs. These are fascinating DNA sequences that have the remarkable ability to move from one location in the genome to another, or sometimes to copy themselves and insert the copy elsewhere. They can jump within the same chromosome or even between different chromosomes.
And they're common.
Incredibly common. They're found in virtually all organisms, from bacteria to humans, and their prevalence is astounding. Get this, nearly half, almost 50% of the entire human genome is derived from transposable elements.
Half. That's way more than the actual genes coding for proteins, right?
Way more. Protein-coding DNA makes up only about 1–2% of our genome. The rest is non-coding, and TEs make up the largest chunk of that.
So what do they do if they jump around that must cause problems?
It certainly can. When a TE inserts into a new location, it can land right in the middle of a gene, disrupting its function. It can alter gene expression if it lands in a regulatory region. Their presence can promote incorrect recombination between different parts of the genome, leading to deletions, duplications, or inversions. They are a significant source of mutation and genome instability.
But they must have some role. Or why would we have so many?
That's the big question. They are often viewed as genomic parasites, but they also play roles in genome evolution and maybe even gene regulation. And scientists have cleverly harnessed them as powerful tools for genetic research, like for creating mutations or tagging genes.
Okay, so how do they move? Are there different kinds?
Yes, two main classes based on their mode of transposition. Class 2 elements are DNA transposons. They move using a cut-and-paste mechanism. They typically have inverted repeats at their ends and encode an enzyme called transposase. The transposase recognizes the ends, cuts the transpose out of its original location, and pastes it into a new target site. They often leave a characteristic small duplication of the target site DNA flanking the inserted element.
Cut and paste. And the famous example is in corn.
Exactly. The ACD system in maize was discovered by Barbara McClintock back in the 1940s and 50s, work that eventually won her the Nobel Prize. She observed how pigment patterns in corn kernels changed in unstable ways. She figured out it was due to mobile elements. The D's dissociation element could cause chromosome breaks or disrupt pigment genes, but it could only move if another element, activator, which provides the transposase, was also present. Her work showed these elements weren't fixed parts of the genome.
Groundbreaking stuff. What's the other class?
Class 1 elements are retrotransposons. These use a copy and paste mechanism that involves an RNA intermediate. They are actually the dominant type in mammals, making up about 42% of the human genome.
Copy and paste via RNA. How does that work?
The retrotransposon DNA is first transcribed into an RNA molecule. This RNA is then used as a template by an enzyme called reverse transcriptase, which the retrotransposon itself often encodes, to make a DNA copy. This DNA copy is then inserted into a new location in the genome by another enzyme, integrase. The key difference is the original retrotransposon stays put, only the copy moves. This allows them to accumulate to very high numbers in the genome. They structurally resemble retroviruses, but typically lack genes needed to form infectious particles.
So they copy themselves all over the place. Any examples?
A classic example is the copia element in Drosophila, fruit flies. Insertion of a copia electrotransposon into an intron of the white gene, which controls eye pigment, can interfere with proper processing of the gene's transcript. This leads to a mutant phenotype called white apricot, with orange-yellow eyes instead of the wild-type red.
Incredible. And you said these make up a huge chunk of our genome. Human Ts.
Yes, the vast majority of Ts in humans are retrotransposons. The main families are lines, long interspersed elements, and sines, short interspersed elements. Together, they account for about 34% of our DNA. Lines are autonomous, encode their own reverse transcriptase, while signs, like the famous ALU elements, are non-autonomous and rely on the line machinery to move.
And do these human Ts actually cause diseases by jumping around?
They absolutely do. Although it's estimated that maybe only 0.2% of new disease-causing mutations in humans are due to TE insertions, there are many documented cases. Line insertions have been found to cause cases of hemophilia, Duchenne muscular dystrophy, and have been implicated in some colon and breast cancers. ALU element, SIAE insertions, are known to be responsible for over 30 different human genetic diseases, including neurofibromatosis, some forms of hemophilia, and even a case where an ALU insertion into the BRCA2 gene contributed to familial breast cancer.
Wow, so these aren't just ancient relics. Some are still active.
Yes, some line elements in particular are still actively transposing in the human genome, though at a low frequency.
And you mentioned that really striking hemophilia case.
Ah, yes, the factor VIII gene case. A boy had hemophilia due to a line insertion disrupting his factor VIII gene on the X chromosome. But when they looked at his mother, the line wasn't on either of her X chromosomes.
So where did it come from?
They eventually found copies of that specific line element on chromosome 22 in both the mother and the father. The inescapable conclusion was that the line element must have jumped from chromosome 22 into the factor VIII gene on the X chromosome in the mother's germ-line, the cells forming her eggs. That mutated X was then passed to her son.
Direct evidence of a jump in a human germ cell. That's mind-blowing.
It really is. It shows these elements are dynamic forces shaping our genomes even now.
So, summing up TEs, they can disrupt genes, cause mutations, rearrange chromosomes. But you also hinted they might play a role in evolution.
Yes, despite their disruptive potential, TEs are increasingly recognized as drivers of genome evolution. Their insertion can create new regulatory sequences, alter gene expression patterns, or even lead to the formation of new genes through shuffling of exons. The repetitive nature of TEs can also promote recombination events, leading to larger-scale genomic rearrangements. And sometimes, parts of TEs can be domesticated or co-opted by the host genome to perform useful functions.
Like the Drosophila telomeres you mentioned.
Exactly. In Drosophila, which lack the usual telomerase enzyme, specific line-like retrotransposons have actually evolved to transpose preferentially to the ends of chromosomes, effectively maintaining chromosome length, the job normally done by telomeres. It's a remarkable example of making something useful out of a genomic parasite.
What an incredible journey. Okay, let's try to wrap this up. So what does this all mean? We've covered gene mutations, defining them, classifying them by change, function, effect, and location. We've seen how they arise spontaneously through replication errors and internal chemistry, and how external agents like chemicals and radiation induce them. We've looked at the real-world impact, like in β-thalassemia and XP, highlighting the vulnerability of our genes. And we've explored the amazing multi-layered DNA repair systems our cells use to constantly fight back against this damage. Plus, the weird and wonderful world of transposable elements.
It really underscores the dynamic nature of the genome. It's not static. It's constantly facing challenges and changes. There's this intricate balance between the forces causing mutation and the sophisticated mechanisms that repair the damage. And the Ts add another layer of complexity, acting as agents of both disruption and potentially evolutionary innovation. These ongoing genetic shifts aren't just errors. They are fundamental processes that generate the variation underlying all biological diversity and adaptation.
So here's a final thought to leave you with. Think about those 60 new mutations you likely inherited, mostly from your father, depending on his age when you were conceived. Consider that your cells right now are detecting and repairing thousands of DNA lesions every single day. This constant microscopic battle for genomic integrity is happening inside you, largely unseen and unfelt. How does this ongoing interplay of damage, repair, and occasional change shape not only who we are as individuals, but also where we might be heading as a species? It's a lot to ponder. Thank you for joining us on this deep dive into the fascinating and sometimes turbulent world of our own genes. Keep exploring.